These are the lecture notes for chapter 20, and we left off on slide 23. So Pasteur boiled broth and then briefly opened the flask that he boiled the broth in. So he sterilized the broth, opened the flask briefly, 89% showed growth. And then he did the same experiment. The first one was done outside the building and the second one inside the building. And the second one, only 32% showed growth. But basically it proved that bacteria or organisms can get in from the air. So he built this neck, this flask with a special neck, and it's open to the air. So he boils the broth to sterilize it, and any bacteria that enter are going to get caught right here, and they're not going to be able to go up the incline. So the broth is going to stay sterile. It's going to remain sterile. 100% has no growth, and that was Louis Pasteur. And we call this process pasteurization. Now, structure of prokaryotes. Remember, prokaryotes have not got a nucleus, but they do have DNA in a nucleoid region. So they lack a membrane-bounded nucleus. They do have an outer cell wall. The bacteria's cell wall, a lot of bacterial cell walls are composed of peptidoglycan. And we make use of that peptidoglycan layer in staining. Some bacteria move using a flagella. Some lack, or bacteria lack membranous organelles. So they're not going to have anything with a membrane like a lysosome or peroxisome or mitochondria or chloroplast. But they may have accessory rings of DNA, so small rings of DNA called plasmids, and a lot of times those plasmids will have an accessory gene located on those plasmids in their nucleic acid structure arrangement. Okay, so prokaryotic cell is going to have cytoplasm. And in the cytoplasm, there's a nucleoid region where the DNA is located. There are ribosomes, which synthesize proteins. There are thylakoids in cyanobacteria because they're the only prokaryotes that can photosynthesize. The cell envelope includes the plasma membrane and the cell wall, and many bacteria also have a glycocalyx for even more protection outside the cell wall. Appendages that can be attached to prokaryotes include flagella to help them swim, a conjugation pilus, which will allow one to exchange DNA with another, and then fimbriae, which can help the prokaryote attach to surfaces. And here are a lot of those structures. So fimbriae are hair-like bristles. The nucleoid region contains a single circular chromosome. It's just bunched up here, but it's really just one single chromosome. If you spread it all out, it would make a circle. The conjugation pilus allows this bacterial cell to donate a plasmid or a ring of DNA to another. The flagellum helps it to swim. Ribosome synthesizes proteins. The blue layer here is the plasma membrane. The red layer is the cell wall. And then the kind of brown, light brown layer is the capsule made up of a glycocalyx. All right, reproduction in prokaryotes is asexual. Prokaryotes reproduce through a process called binary fission, and the generation time, in other words, the time it takes one bacterium to divide into two, is as short as 12 minutes. It can be longer, but it's as short as 12 minutes. It's important for you to know that prokaryotes are haploid, so we can't have heterozygotes because each bacterium only gets one copy of each gene. They don't have two parents. Some bacteria form resistant endospores under unfavorable conditions, like maybe it's too hot or too dry, and or the pH isn't where it needs to be. Then they'll form these endospores until conditions become favorable. Now, this is a picture of a bacterial cell that's in the process of dividing by binary fission. Now, there are 3 ways that bacteria can exchange, or can their genes can recombine. We call that genetic recombination. So we can get varieties, different varieties. Not only do bacteria mutate at a very fast rate, their genes will mutate very fast compared to eukaryotic cells, but also they can carry out this process called conjugation. One cell, called the donor cell, will line up with the recipient cell and inject a plasmid, which is just a small circular piece of DNA, inject that plasmid into the recipient cell. Transformation occurs, and Frederick Griffith is the one that discovered this. We mentioned it, I think, at some point. But it occurs when a bacterial cell that is alive picks up pieces of DNA from the environment, and that could be from other dead prokaryotes, and that DNA becomes incorporated into the genome of the living bacterium. Then we have transduction, and that is when bacteriophages, remember those are viruses that infect bacteria, actually carry portions of bacterial DNA from one cell to another, and in that way the viruses serve as vectors. So that's transduction. So conjugation involves a conjugation pilus, a donor cell, and a recipient cell. Transformation involves extra DNA in the environment, oftentimes in the form of dead bacteria. Transduction involves viruses. Now, section 20.3 on bacteria. They are the most common type of prokaryote. Most bacterial cells have peptidoglycan in their cell wall. Peptidoglycan. You say that with me. Peptidoglycan. Peptidoglycan. Bacteria are commonly differentiated using the Gram stain procedure. You're going to do that in our virtual lab. When washed after staining, Gram-positive bacteria retain a violet-colored dye and appear dark purple under the microscope, and Gram-negative bacteria have a thinner layer of peptidoglycan, so they don't retain that purple dye. They appear, it says red, but if you look in real life, they actually look pink. Gram-negative bacteria have a second plasma membrane, which blocks antibiotic drugs, so that makes infections difficult to treat. Gram-negative infections tend to be difficult to treat. But even though they have a second plasma membrane, they only have a very thin peptidoglycan layer, whereas the Gram-positive have a very thick peptidoglycan layer. And this is a picture of how Gram staining works. You've got a sample of bacteria on a glass slide, and you add purple stain to it, and then you add iodine, which helps to hold the stain in the chemicals that make up the cell wall. Then you use a decolorizer, and what that does is removes the stain from the glass but leaves it on the bacteria. Because if you stain the whole glass purple and you don't remove that stain off of that glass, all you see is purple. You're not going to see the bacteria. The role of staining is so you can see the bacteria. Then we're going to do another type of stain called safranine, and that is a red stain, but under the microscope it makes these bacteria appear pink. So the Gram-pink bacteria, we call the pink bacteria Gram-negative, and we call the purple bacteria Gram-positive. And that's one way to start identifying bacteria. Now, you can also use shapes. I can see some shapes here that are showing up in picture B down at the bottom. I can see some shapes. They look like bacillus, which is rod-shaped. And here the shapes are listed. So the bacteria can be spiral. We call those spirilla or spirillum. They can be rod-shaped or oval. We call those bacilli or bacillus. They can be round like a basketball. We call those cocci or coccus. And so here's a spirillum, here's a bacillus, and here is a chain of coccus, which would be streptococcus. Strepto means a chain of. All right, now metabolism in bacteria is more, there's more varieties of metabolism than there are in some of the more complex organisms like plants and animals. So bacteria can be obligate aerobes. An aerobe requires oxygen. In other words, it must have oxygen to live. We, humans, are obligate aerobes, unable to grow in the absence of free oxygen. Then there's obligate anaerobes. These are unable to grow in the presence of free oxygen. So obligate anaerobes can't grow in the presence of oxygen. They will die. Oxygen will kill them. Examples of obligate anaerobes are the bacteria that causes botulism, gas gangrene, and tetanus. Then we have facultative anaerobes, and these are either able to grow in the presence of oxygen or the absence of oxygen. They can grow either way. Now, autotrophic means that they make their own food. They don't consume their food, they make it themselves. Photoautotrophs carry out photosynthesis, but the anoxygenic green sulfur and purple bacteria that live in oxygen-poor conditions, they don't actually produce oxygen. And then there's the oxygenic ones like the cyanobacteria. But what photoautotrophs do, they use solar energy or sunlight to reduce or change carbon dioxide to sugar, organic compounds like sugars and starches, oils. Then we have chemoautotrophs. And these still make their own food, but they use a different source of energy than the sun. They use inorganic compounds. They break them down to obtain energy, and then they use that energy to reduce carbon dioxide to an organic compound. Chemosynthetic, these are bacteria that live in environments such as deep sea vents 2.5 kilometers below sea level. And since we don't deal with kilometers much, and I don't know it by heart, let's see, how many kilometers in a mile? 1 mile. So a kilometer is bigger than a mile. It's 1.6 kilometers in a single mile. And 1 mile is 1.6 kilometers. Okay. All right. Photoautotrophic bacteria can be anoxygenic. That means they don't produce oxygen. They only use photosystem 1. They don't use photosystem 2. They have a unique type of chlorophyll called bacteriochlorophyll. And then we have oxygenic photosynthesis like plants do. These bacteria produce oxygen during the process of photosynthesis, and they contain photosystems 1 and 2. Symbiosis. Mutualism means both organisms benefit. Commensalism means one benefits and the other is neither harmed nor benefited. Parasitism is when one benefits and the other is harmed. Up here at the top, we have chemoheterotrophs, which are prokaryotes or any organism really that takes in organic nutrients and breaks them down for energy. Saprotrophs are decomposers. Generally, that's going to be your bacteria and your fungi. And bacteria can be free-living or symbiotic, and your 3 types of symbiosis is here.